This video is going to go through the entire transformation experiment, and then the last video will be going through the results. So this is the guide that will be used to go through all of the steps of the experiment. The two tubes, now in the video prior to that, there was a light blue tube and a pink tube. You would go through and label one tube positive, one tube negative. And these are these micro tubes. Sometimes they're called microcentrifuge tubes. You would label one a plus, one a minus. During this step, you would add what is called the transformation solution. It was labeled as a T on the other video. You would basically divide up what was in the tube that was given to you, put half in the plus, half in the minus. Place the tubes on crushed ice. Now after you add this is the calcium chloride. You would close the tubes, place the tubes on ice. Now, while these are sitting on ice, you would pick up the little starter plate that had the E. coli cells that I had described earlier. You pick up one colony, put it in one in the other. Like it says here, you would basically take the loop, you're going to pick up a colony, and you're going to transfer that colony into each of the two tubes. I think that is the plus one there. It says basically take them off the ice, put some bacteria inside the tube. And I know it says spin that around. Place the tube back on the ice. Use the new sterile tube and repeat for the negative tube. Now in the video you're going to watch, they're actually heating the loop between. They're not using the plastic pipettes. Or the plastic loops. Alright, now this step, so you're going to put these back on the ice. Okay, once you add the bacteria, you put them back on the ice again. Now you're going to pick up the little bottle that has the plasmid DNA. It actually comes dry. You add some transformation solution. It will then rehydrate it, and you're only going to put these little plasmids into the plus tube. That's why you have it labeled as a plus. Do not put it in the negative. So now you have some cells that are going to be exposed to the plasmid, and you have some cells that are not. And what you would do next is once you add the plasmid to your plus only, like it says here, incubate the tubes on ice for 10 minutes. That's the exciting part. During that time while you're waiting, you can go ahead and label. You would each be giving, as a group, 4 of these little micro plates. You would label them just like this. And if you saw the video prior to this, here's the 2 LB ampicillin. Here's the LB ampicillin arabinose, and then here is the LB plate. Now, after 10 minutes of sitting on ice, you would then take your little styrofoam boat with the 2 tubes in there, and you're going to float it in a water bath. And it says there, and I said in the previous video 30 seconds. It says 50 seconds here. Sorry, I was off. But basically, you take them from the ice, you put them in the water bath for 50 seconds, and then you put them back on ice. That is called the heat shock method. And here's the thing. This transformation solution that you've added during this step, it's designed to make the cell wall and the cell membrane more competent. That means more permeable to accepting large molecules like these plasmids. When you then put it on the ice, the heat back to the ice, that's called heat shock. It's a procedure that forces the plasmids in the solution inside the cell through the permeable openings, hopefully. Now, one of the last steps is that you would then take what is called the Luria broth, which was the LB in the previous video. You're going to divide it up and put half in one, half in the other. This is for growth. This is hopefully to stimulate growth of the new cells so they can then start expressing the genes on the plasmid in the plus vial. And it says here, let it incubate at room temp for 10 minutes. E. coli divides every 20 minutes, so that's really not that long of a time. Now, once the 10 minutes have passed, you would then take, it says 100 microns, or you could just take a loop, and you would then spread the cells out on each of the plates. So here's the positive P-glow, then the positive LB, AMP, ARA plate, the 2 positive plates, and the 2 negative plates. And you can see how they're labeled. You're going to have a plus LB, AMP, a plus LB, AMP, ARA, AMP stands for ampicillin. So all of these plates here, all three of them have ampicillin in the agar. Only this one has arabinose as well. So it's got three things in it, only that one. And this is just a general media plate for growth. You can see by the, you spread it around as best you can. You would then stack the plates like this, and then you would put them into an incubator. So please watch the video next on how to conduct the experiment.
In this video, you will learn how to perform a bacterial transformation. E. coli HB101 bacteria will be transformed with P-glow plasmid using a calcium chloride transformation. To start this activity, you will need an LB agar starter plate streaked for single colonies of E. coli HB101 and 2 microcentrifuge tubes, each with 250 microliters of 0.05 molar calcium chloride or transformation solution. One of the tubes should be labeled plus and the other minus. Both tubes should be kept on ice. Flame an inoculation loop and allow it to cool. You can lightly touch the loop on an empty spot on the starter plate to make certain the loop is cool. Then gently scrape 1 large or 2 small colonies from the surface of the starter plate so that they can collect on the loop. Transfer the loop with bacteria to the plus tube and swirl it in the transformation solution to disperse the bacteria. Close the tube and place it back on ice. Flame the loop and repeat the process to add bacteria to the minus tube. Using a micropipette set to 10 microliters, obtain 10 microliters of P-glow plasmid. The volume of plasmid you will be working with is usually very small, so make certain you are looking directly into the sample as you pipette. You should also visually inspect your pipette tip to verify you have obtained 10 microliters of sample. Pipette the 10 microliters of plasmid into the plus tube. Do not add any plasmid into the minus tube. Incubate both tubes on ice for 10 minutes, making sure the tubes are in full contact with the ice. During the 10-minute wait, using a waterproof marker, label your LB agar plates according to the instructions provided with your lab protocol. After your tubes have been on ice for 10 minutes, transfer both the plus and the minus tube directly from the ice into a 42°C water bath for exactly 50 seconds. Then immediately place them back on ice. Incubation at 42°C is used to heat shock the bacteria. Incubate the tubes for 2 minutes on ice. After 2 minutes, remove the tubes from ice. Using either an adjustable or disposable pipette, transfer 250 microliters of LB broth into the plus tube using aseptic technique. Repeat this process to add LB broth to the minus tube, and then incubate both tubes for 10 minutes at room temperature. Using an adjustable or disposable pipette, transfer 100 microliters of each transformation mixture onto the appropriately labeled LB agar plate using a sterile pipette or pipette tip each time. Flame the inoculation loop and let it cool down. Spread the bacteria over the entire surface of the plate in all directions. Rotating the plate a quarter turn between spreads will ensure the bacteria is spread evenly across the plate. When finished, flame the loop and repeat this process for each plate. Turn the plates with the bottom or gel side facing up and stack the plates. Place the plates in a 37 degrees Celsius incubator for 16 to 24 hours.